In this video, we're going to talk about the second messenger system. Now, we're going to look at the system in terms of generalities. We're not going to mention the specific enzymes that get turned on or off. Let's start with an overview. Why does this system exist? Well, just a reminder is that when you're looking at the cell membrane, right, we have hydrophilic areas, and then we have hydrophobic areas. So only substances of a certain type of chemistry can physically cross into the cell. And we're talking, say, messengers that want to come into the cell and basically tell the cell to do something. It could be turning on an enzyme or to turn on a metabolic pathway. So some substances can come straight into the cell, but many of them can't because of their chemical nature. In this case, if they did try to enter the cell, they can't. They're just refused entry. So this is a system that will allow those type of chemicals to transfer the message to the inside of the cell. And it's called second messenger because the first messenger will be the actual chemical that's coming up to the surface of the cell. Let's say it could be a hormone, one of the hormones that cannot get directly into the cell. And then what it's going to do is it's going to transfer the message through a number of proteins, activate an enzyme, which will then create a substance, which is like a surrogate for the messenger. It's basically carrying the message indirectly. Now let's walk through it. It's a multi-step process. So the first thing you need, now the example, before I go too far, the example they're using here is epinephrine, and that's this red triangle. So epinephrine cannot directly get through the cell, can't do it. So what happens is the epinephrine will bind onto a specific receptor. There's no general receptor that will deal with every message. So every message has its own receptor. It's very much like a lock and a key fit. So if epinephrine comes up to the cell and does find the corresponding receptor and binds onto it, then what happens is, now first of all, this message cannot enter the cell. It won't enter the cell. It has to stay on the outside. But what happens with the binding of the first messenger with the receptor, like lock and key fit, that causes the activation of something called the G protein. Now, once the G protein is activated, the G protein then interacts with an enzyme, adenylate cyclase. Look at the name, A's. Tells us it's an enzyme. Look at the first one, adenylate. That's going to sound somewhat familiar when we see what it actually works on. So let's just quickly review up to this point. First messenger can't enter the cell. Binds onto a specific receptor. The receptor then leads to the activation of the G protein. The G protein, once it's activated, will interact with the adenylate cyclase. Now, this is an enzyme that's just now being activated by the G protein. The adenylate cyclase will take one ATP, and as an enzyme, it's now going to pull off one phosphate, two phosphates. So it's just pulled off two phosphates. Now, of course, if I've taken off two phosphates, I've gone from ATP then to ADP, because that's removing one phosphate, and then I take off one more phosphate, and I end up with AMP. And remember in the previous chapter, we said that AMP often will, the phosphate group will actually bind in sort of a circular way back to the other part of the molecule. And then we give it a special name, and I'm going to put the letter C in front. And that becomes cyclic AMP. So the messenger bound onto the receptor like a lock and a key. The receptor activated the G protein. The activated G protein activated the adenylate cyclase. The adenylate cyclase took an ATP and pulled off two phosphates. That produces cyclic AMP. Now, the cyclic AMP, we call it the second messenger. Okay. Now it's not the original epinephrine. They're very different things. But what's happened is it's in many ways it's a surrogate for this. So through this process, we've activated the second messenger, cyclic AMP. Now, what does cyclic AMP do? Cyclic AMP, it'll take an inactive kinase, and if you remember, I'm just going to make this larger, from the previous chapter, we talked what kinases do. And kinases do phosphorylation. So the cyclic AMP, it takes an inactive kinase and it activates it. The activated kinase, and remember, the job of a kinase is to take a phosphate and attach it onto molecules, basically macromolecules. So the activated kinase then adds a phosphate to an inactive enzyme, and now that enzyme is activated. Now, what has all this done? It seems like a lot of steps, but this has now, since these enzymes are now activated, they've always been in the cell. They've been inactivated. Now they've been turned on. They can go and start the various metabolic processes. So this is a way, second messenger system is a way for messages that cannot directly enter the cell to indirectly send their message to us and create a second messenger, and its job is to take an inactive kinase, activate the kinase, and this activated kinase, as kinases do, it phosphorylates. It phosphorylates an inactive enzyme. I have an activated enzyme now, and then I've turned on a metabolic pathway. Okay. Now, just a little side note is that the first messenger, of course, we don't want it to stick around here forever because it'll keep activating and keep just trying to reactivate the receptor. And in previous slides, we talked about membrane receptors. Membrane enzymes. And certainly what happens in this case, some of these enzymes are embedded there, and what their job is to basically break down the messenger once it detaches and starts to float around. Or if there's excess, these embedded enzymes will basically inactivate the first messenger once it's done its job. Okay, that's a bit of a quick walkthrough. This is a very important system. So I did also include on our webpage a little video that also describes the second messenger system. Feel free to have it, read through your textbook, and certainly if you have any questions, let me know in our Zoom meetings.